Hello, in this video we're going to be talking about osmosis. So let's not waste any of your time. Let's get right to it. Now when we're talking osmosis, again for the third time, we want to emphasize those passive transport principles that no energy is required, okay, and we're moving with the concentration gradient. In other words, wherever there's a bunch, we're going to move to areas that they're not. In this case, we're crossing the membrane, so if there's a bunch on the outside and not much on the inside, then we're going to move on to the inside, or the opposite may be true. Now the thing about osmosis is it is trying to dilute. So we're not moving water from areas to where it's not to areas, to where it is to where it's not. We're not focused on moving. Osmosis does not focus on moving water to balance out the amounts of water. Osmosis is trying to dilute, okay, to trying to make the amount of the solvent to be an equal amount on each side of the membrane. So it's all about where is a bunch of solvent. We're going to go there to try to dilute this down. The way to think about osmosis, and we're going to look at lots of examples to try to help grasp that concept. But when we're not moving water for the sake of moving water, we're moving water with a purpose to make the equilibrium rates based on how much dissolved substance. So for example, if there was just a little bit of salt on the inside of a cell and there was a whole bunch of salt on the outside of the cell, then the water is going to move to the outside to try to dilute that down. Okay, that's the process that occurs when you pour salt on the slug and it simply dissolves in front of you, in front of your eyes, because all of its water is leaving the cells to try to dilute that excess salt down. And there are some organisms that have to try to combat this because of the conditions they live in. Think about an ocean, a fish that's in the ocean. It's got lots of salt all around it. So what is its water going to want to do? It's going to want to leave the cells and go dilute all that salt. But if that happens, okay, then the cells could shrink and die. So in that instance, an organism has to make sure it takes in plenty of water to replace that water, because it's a constant osmosis movement out. Think about a freshwater fish. Okay, it doesn't live in a salty environment, and the salt it has in its cells is going to attract or bring in water. So it's got to really be cognizant of that. But it's the idea of the water moving for the sake of a purpose, to get a job done, to dilute down. So think of firemen. Okay, when a fireman attaches its hose to the fire hydrant, is it looking to move the water just for the sake of moving water? No, it's trying to put out the fire. So it's going to move the water towards the fire, trying to extinguish it. The same thing with osmosis. The water is moving to try to dilute down the solvent, whatever it might be. Think about the military as well. Okay, in response to a threat, do they just move the military to another place just simply because to move the military? No, they move to where a threat is. There's a threat somewhere, then they move to that area to deal with that threat. And that's the same idea with water and osmosis. Okay, so here's an example. You can see the picture beside me. I got it's before and after. Okay, so it's the same beaker, just before and after. In the first picture, the one closest to me, whatever the solvent might be, salt, potassium, oxygen gas, whatever it might be. There's a whole bunch on the left, on the right, I mean, and there's a little bit on the left. Now the solvent can't move across that membrane, which is the red dotted line, but water can because of diffusion. So what the water's going to try to do is some of the water, because on the water on the left-hand side, that's about 90-95% water and 5 to 10% solvent. But on the other side, it may be 60% solvent, 40% water, okay, something like that. And so the water is going to move to try to balance it out, okay, dissolve it, dilute it down. So when the water moves, now we've got 50% water, 50% solvent on each side. So the water did not move, okay, for the sake of the water, because actually in the first picture, the water is 50-50, but that's not what water is involved in. Model's not thinking about that. It's moving to an area of greater solvent concentration. So if you want to know where the water of the cell is going to go, or the water around the cell, find the solvent amounts, and that will tell you where the water is going to go. Okay, now there are various types of situations in which osmosis is involved. Hypertonic, hypotonic, isotonic. The first two, hypertonic, hypotonic, for an animal cell, those are potentially bad situations. But it's super important that you recognize what each of them are and what the desired, or what the output is going to be. Okay, so a hypertonic situation, that means there's more solvent outside the cell. Okay, so the water is going to leave the cell. There's more salt on the outside of the cell, so the water is going to leave. That's a hypertonic solution. Hypertonic is going to refer to that there's a lower solvent amount inside the cell than outside. Then the way I always remind students to think about this, what do you do with a hyperactive kid? You send them outside to play. Okay, the water leaves the cell. Hypotonic is the exact opposite. Now there's more solvent inside the cell than there is outside the cell. So the water is going to come in. And the way you can think about this is that O in hypotonic, in the past, things that we've done to help remember this is we've written that word out and we made the O really big, okay, and made it into a pond. If you know anything about ponds, especially on a farm, the ponds typically are the lowest ground elevation, and the water runs into it. That's how it gets filled initially or naturally. And so we draw that O as a pond to let us know the water is coming into the cell. Okay, so a hypertonic solution, there's more solvent inside the cell. The water is going to run in. And a hypertonic, there's more solvent outside the cell, so the water is going to leave. Okay. Now the other one, the third one, is the one that our cells have to be in or need to be in, and that's isotonic. In other words, it doesn't mean, the definition means no movement, and that's not accurate. What it means is equal movement. The amount that's coming out needs to also come in, so it's balanced at all times. One important aspect of this is an IV. Okay, you're going to get an IV, especially if you're going to be put to sleep with the IV for the idea of a surgery or part of the anesthesia is being delivered in that manner. A lot of times they'll take a blood sample and test it to see what your cell levels are, because that IV has to be isotonic. Excuse me. if it's not isotonic and they give you a hypertonic IV, which means lower solvent amounts, and then the water then rushes. Lower, so the water rushes out. You don't want that. It doesn't even be a hypotonic, water rushing into your cells. That's bad. So it has to be isotonic. Now these next two screens are going to show examples of what we're talking about here. Okay, so this is an animal cell. These are obviously even blood cells, perhaps. And you can see that hypertonic going outside. The water is going to go outside the cell, and you can see the cells shriveled up, which is bad. And the hypotonic on the far right, again, hypo, water running into the ponds, hypotonic running into the cells, and you can even see one cell bursting there. So whether they shrink and lyse or whether they swell and burst, neither are good for the cells. The middle ones where we have to be isotonic towards equal amounts coming in and going out, and everything is well. Okay. Now, plant cells respond differently to than our cells do. Hypertonic. Nobody likes a hypertonic because the water leaves the cell, and that's bad for a plant. Plants could die pretty quickly. Of course, we could too. But when it comes to hypotonic, for us, isotonic was the best. For plants, hypotonic is the best. And the reason why is because plant cells have a central vacuole. Okay, they've got that one big vacuole in the cell, and it likes to be full of water, because when it does, that water presses against the cell wall. And when that happens, okay, just like our vertebrae, those cells become now very firm and hard, rigid, to where almost like a brick now. And if you take you take bricks and stack them up, okay, you can build a little something. If you think about a blade of grass, if the cells are all full of water in their vacuoles and they're pressed up against the cell wall, now what you can do is as those cells are stacked naturally, you can create like a spine or a stack of Legos or bricks or whatever, to where the plant can stand upright. When you see a plant wilting over, we all know that's an obvious sign of lack of water, and this is the reason why. The pressure has reduced, the cell wall isn't as firm and rigid now, and therefore the plant begins to lean over. But if the plant's not been damaged, then you can add some water to it, and it'll perk right up and stand right back up in just a short amount of time. But that pressure is called turgor pressure. Okay, turgor pressure, when the vacuole is full and it's pressing against the cell wall. Okay, that turgor pressure that we just spoke about comes about from osmotic pressure, or pressure created by osmosis. Cells in a hypotonic, okay, causing it to swell, creates pressure. And if the membrane is strong enough, as we see with plant cells, then it can handle that. But like ours, okay, it cannot handle that. Animal cells, we've got to be in an isotonic situation, or it's just going to end badly for us. So that's why the doctors and in the medical field take such big precautions on that. Now, there are some things that organisms can do to help kind of balance out this pressure. Contractual vacuoles is one of those ways. We spoke about these when we talked about organelles and the three types of vacuoles: contractual, central, and in storage, like what we have. But there are some organisms that has those contractual vacuoles, and when the water pressure gets too high, okay, they can just shoot out some, squirt out some, blow out some, whatever you want to call it, to help adjust that fluid. But it's that whole idea of osmotic pressure, hypertonic, hypotonic, that fish, as I mentioned, in watery environments have to be concerned about, or any organism in a watery environment. If you're in salt water, you've got to deal with that because the water from your cells wants to leave. If you are freshwater, then the water wants to rush in, and it's got to take care, got to make sure it handles that situation as well. So osmosis pressure, again, our osmosis, the main concept is to realize is that the water is not moving for the sake of water. It is moving to dilute down, okay, a collection of solvent. Well, that's our video over osmosis. The next time we get together, we're going to be talking about active transport. So we'll see you then.